Anderson is the Bradley R. Dewey Scholarship Award recipient for this year, and it is the tradition that the recipient of this award gives a common hour address. I'm pleased to say that I knew the late Brad Dewey. Brad always wanted us to hire the very best faculty, and that is something we continue to do and will continue to do here at Franklin Marshall College. I'm also pleased to acknowledge that Marsha Dewey joins us today. The Bradley R. Dewey Scholarship Award recognizes the faculty member who best exemplifies the ideal of the scholar whose research efforts reflect and inspire excellence and enlighten teaching. And the recipient this year is Michael Anderson, Associate Professor of Psychology and Chair of our program in Scientific and Philosophical Studies of Mind. He was recently described both as one of the most creative neuroscientists of his generation and also the world's leading philosopher of neuroscience, a pair of compliments indicative of the sort of interdisciplinary impact that we cultivate here at Franklin Marshall College. Not surprisingly, Professor Anderson's remarkable intellectual breadth is well grounded in the liberal arts, including a PhD in philosophy from Yale, a BS in pre-medical studies from Notre Dame, and extensive postdoctoral training in computer science and artificial intelligence at the University of Maryland. When he came to F&M in 2006, Professor Anderson had already begun his groundbreaking work with a highly influential paper entitled Embodied Cognition, which has garnered more than 800 citations and is being used in classes around the world. Since then, his scholarship has begun to open a new paradigm for understanding the mind and the brain. In his book, After Phenology, Anderson uses, which came out in 2014, Anderson uses cutting-edge evidence to outline a new approach to understanding the brain, which we're all eager to hear about today. With data from thousands of neuroimaging studies, he discovered new forms of neuroplasticity, which help build neural coalitions that flexibly serve multiple functions. Anderson links this plasticity with a deeper account of how organisms evolved increasingly flexible abilities to interact with a complex, unpredictable world. The result is a new account of the mind as biologically grounded on the evolution of complex action. His influence has continued to grow at F&M with more than 100 publications, including groundbreaking and widely read articles in the most prestigious journals, and postdoc or post-tenure fellowships at Stanford University, where he spent writing After Phenology. This book has garnered tremendous interest. It is the book of the year and beyond in his field. It's receiving extraordinary attention. Professor Anderson's creative energy has also been well engaged with our students at all levels, from first-year seminars to senior capstones and courses that are technical, historical, and philosophical. He has mentored numerous students in independent research, with many earning departmental honors and going on to prestigious graduate programs. He's renowned for challenging his students to appreciate past insights and to imagine the possibility of reconfiguring one's perspective and understanding how to create new knowledge. Michael Anderson truly exemplifies the high ideals advanced by all of our faculty, and in particular by those who received the Dewey Award. I know some of his friends and many of his departmental colleagues are here today, and I'm sure they're very proud of him. So is the college. Franklin Marshall is very pleased to recognize his extraordinary contributions to scholarship and education, and we're all grateful that he will share his exciting research now in this Common Hour address. Please welcome Dr. Michael Anderson.
Thank you for that introduction, and thank you everybody for coming. I hope you're enjoying your pizza. I'm going to tell you some things about the brain and about how we learn these things about the brain. The outline of the talk is pretty simple. Tell you about the functional organization of the brain, how the thing works, how it's organized, and then tell you about how we know that, because being a scientist, I don't want to just tell you things, I want to show you things, and I want to see you, have you appreciate how we figured these things out. And then, so we'll talk about just a couple kinds of analyses that I've done that help us understand how the brain works, how it's organized. And then the third part of the talk is really what it means, right? What it means for evolution, what it means for understanding who we are and how we work. So, first part. This is what you're used to seeing, right? The media loves this kind of stuff. You see a brain image, and you see the area for X, the area for language, the area for your love of your cell phone, the area for anything under the sun, okay? And it's true that when you look at one study at a time, one neuroimaging study at a time, that's kind of how it looks, right? It looks like the brain is specialized for one thing or this, by the way, this kills me. That's the language area active in a typical 3-year-old when hearing a story. But we do this all the time. So we have the face area, the place area, the object area, the body area, right? As if each part of the brain is dedicated to a very particular thing. And again, as I say, when you do one study, that's kind of how it looks. Leaves the impression of specialization, but this is where this leads. Right? This is a form of phrenology. This is a form of reading the bumps on the skull and figuring there's some kind of character trait you can find out underneath. We all do have a donut area, by the way. That's true. But for the rest of it, this is not true. So I say, the main thing that I've done, and it's sort of an obvious thing to have done, frankly, is, well, instead of looking at one study at a time, let's look at everything all at the same time. Right? Then we can start to see something very different emerge. So here's what you discover, and I'll tell you how we figured these things out in a second. You discover, when you look at all the studies at once, that the mapping between cognitive functions and brain areas is many to many. That is, for any cognitive function, it takes many parts of the brain, and any part of the brain is involved in many different cognitive functions. So individual regions turn out to be used in diverse tasks across many task categories. And how you get differential function, right, how you can do one thing rather than another, is not by activating a particular part of your brain versus a different part of your brain. It's by activating the same parts of your brain in different kinds of functional coalitions. Right? So it's really all about how the parts of the brain cooperate under different conditions. And I'll show you again how we figured that out. So how do we know? Well, the first thing that I want to tell you about is a functional diversity analysis. Here's how you do those in 3 easy steps. Step 1, you build a large collection, right now it's over 6,000 experiments of brain imaging data. So, by the way, this is the thing that I tortured my students here with. They're the ones who had to sit in my lab hour after hour, copying and collating and putting data into a big database, but it was completely worth it for me. Then you categorize the cognitive processes that were examined under those experimental conditions. So you have emotion tasks, or visual perception tasks, or auditory perception tasks, and so on. And then you calculate the diversity of functions observed in each part of the brain. What does that mean? Don't freak out, it's just an equation. Right? Here's what we did. We said, well, somebody must have thought of this kind of thing before. Who would that be? Well, it turns out it's ecologists, because ecologists are very interested in the diversity of ecosystems. And this is the equation they use. Don't worry about it. I'll make it simple in a second. They ask things like, how diverse are the species in a particular ecological niche? And they use this to do it. And so if in a particular part of the planet you have many plants and animals from many different species, you have very high diversity. That is a diversity that's close to 1. If in a particular part of the ecosystem, like the front lawn, you have very few species in very few categories, that's going to give you diversity closer to 0. So all we did was adapt this to cognitive functions and categories. Right? So we're going to look at each part of the brain, each location in the brain, and say how many different cognitive functions and how many different categories does that part of the brain support. Okay, now here comes the data part. But for you to understand the data and appreciate the data, you need to know how to interpret the data. So this is a crash course in interpreting histograms. A histogram is just a way of looking at the number of measurements you take and what the value of those measurements are, right? So you take a measurement, close to 0, and you put it on your graph. You take another measurement, I don't know, maybe 0.5, put it on your graph. Take another measurement, right? You see how this goes. So every measurement, you place it on the graph, and that gives you a sort of characteristic shape to your data. And the shape of this data tells you something about the system you're looking at, the system you're measuring. Okay? So here's one that's near and dear to the hearts of many here. This is the histogram for SAT scores in the United States. All right, so you have a nice normal distribution. Around about 1,500-ish is the median score, the mean score. Right, so that's a very large histogram. We're talking about hundreds of thousands of measurements there. Here's a different histogram. This is the distribution of guesses of how much money's in Brad Pitt's wallet. This organizes itself around about $500. I guess that you figure he was rich, so he must carry more money in his wallet. Here's a distribution, a histogram of U.S. income. This is a very different distribution, right? You can see from this one, the way it's skewed, that most people don't earn very much money, and a few people earn a whole lot of money. This is a distribution of a typical F&M test score in a class, right? We're all very smart here, so most of our scores are way high. Although once in a while there's, that student. Nobody here, of course, because they wouldn't come. Anyway, I hope you get the point, right? So so a brain with very specialized regions, right? The way things look when you do a single study at a time, will have a histogram probably looks something like this, right? Most scores down on the lower end of the scale. And a brain that's not very specialized, right? That's characterized by high diversity might look more like this, right? With things skewed up towards the high end of the scale. So what I did is I showed you the equation, and what I did, I looked at with, I think this particular study was about 3,000 neuroimaging studies. I looked at every location in the brain, right? So every 2 millimeter by 2 millimeter by 2 millimeter part of the brain, so-called voxel, right? A volumetric pixel. So every voxel in the brain measured the diversity at that location. So it gives you thousands of measurements. Here are the data. Right? So you see things skewed way up towards 1. There isn't actually much in the brain that's, my laser pointer doesn't really work here. There isn't actually much in the brain that's totally, does everything, right? It's differentiated, but you can see that it's mostly skewed towards the high end of the scale, right? Parts of the brain do lots of different things under lots of different circumstances. Right? So that's how we figured that part out. Diving into these data a little bit deeper, these are called spider plots. You can see why, I guess. The way you read a spider plot is the distance out from the center indicates the relative proportion of activations under each condition. So in this particular case, I looked at 11 different categories of tasks, so emotion, action, different kinds of language tasks, different kinds of memory tasks, and so on. That 0.88, it's pretty high diversity there. You can see that part of the brain, that's part of the anterior insula, for those who care, that's involved in pretty much everything except vision, to some degree or other. And there are a few relative specialists. So that 0.41 diversity, that's really, you can see down the low end of the scale, there's not much of the brain looks like that. That's a part of auditory cortex right here. And that's mostly dedicated to hearing things. Right, it does a couple other stuff, a little bit of other things, but mostly it's dedicated to hearing things. But very little of the brain is like that. A typical brain region looks like that 0.76. That's about the median of these data. That's a region of the thalamus. And you can see that it does lots of different things. And one of the things that is going to come back about this is that different parts of the brain, they're all very diverse, right? But they're differently diverse from one another, right? So you don't get functional specialization in the way we normally think about it, but you do get functional differentiation. Right? One part of the brain is not like another part of the brain. So they're doing different things. They're capable of different things. And because they're capable of different things, right, just as you or I and everybody else, right, you're capable of lots of different things, and so you do lots of different things. But you don't do everything, right? Because there are certain things you can't do or don't like to do. Same thing for any part of the brain. This is just projecting those values onto a picture of the brain because the Neuroscientists Union requires you to show pictures of the brain when you give talks on the brain. And so I'm fulfilling my obligation, Neuroscientists Union. This is a cool hot scale, right? You can see that things are quite diverse across the brain. I'll show you different views here. Let me show you one other way of looking at these. This is so-called Z-scored. So this is looking at things that are greater than average versus less than average diversity. And what I like about this particular view is it shows you that there is differences in diversity too. And you can start asking questions. I won't get into this today. But you can start asking questions about why this region is really diverse, much more than average, and why this other region is much less than average diversity, right? So this opens up a whole area of research just in looking at the relative diversity of different areas and trying to figure out why that is and what that means. Okay, so this is back to the point I made earlier. Functional differentiation is not the same as functional specialization. So these are spider plots using many more domains than the ones I showed you before, so a bit more complicated. I think there's 20 some odd domains here. These are regions of left and right anterior insula. With some colleagues I did of sort of in-depth analysis of that particular part of the brain. And you can see, even in the same section of the brain, right, the three subdivisions of that section look very different from one another functionally. Right? So they're responsible for a different range of tasks. And again, each part of the brain has its sort of characteristic profile. We call these functional fingerprints. Right? So these functional fingerprints differ from place to place in the brain. But they're all pretty much fairly complex and tell us something about what it is that part of the brain can do. So for instance, turns out, parts of your brain involved in sensory awareness of your fingers are also involved in mathematics tasks, ordering tasks, all things like that. Turns out, parts of your brain involved in motor control are also involved in abstract planning, language, and more stuff. And we'll get back to this point near the end of the talk, because this tells us something important about how it is that we've achieved human-level intelligence with some essentially primate brains. Okay. The other thing I want to show you data-wise is functional connectivity analysis. Why do we do that? Well, as I said earlier, we get differential function in the brain by putting together the pieces in different coalitions. So is that really true? In order to figure that out, we do a functional connectivity analysis. So we look for evidence of statistical dependence in the activity of different brain regions. That is, if you see activity in region A, does that mean activity in region B is therefore more likely? Right? And that would indicate there's a kind of a functional partnership going on. There's some kind of functional connection. So we do that. So if so, we say the regions are functionally connected. They act as partners for a given task. And the results of these analyses, you represent these as a graph. Another crash course. What's a graph? A graph is just a set of nodes joined by edges, where the nodes represent something and the edges represent a relationship. Now, obviously, our nodes are going to be brain areas, and the edges are going to be functional connections. But they're all different kinds of graphs that you can look at. For instance, airline route maps. In an airline route map, the nodes are airports or cities, and the edges indicate flights between those cities. Right? So this is a fragment of the delta graph, as anyone who's flown through Atlanta can figure out. There also can be social networks, right? Now the nodes are people or accounts, and edges indicate friendship, whatever that means these days, right? So this is a fragment of a social network. This is a brain network. The nodes here are brain areas, and the edges indicate, in this case, physical connectivity. Right, so this is looking at how densely interconnected parts of the brain are. Okay, here are my nodes. You have to use a little imagination here. Each sphere here represents a part of the brain, and we're looking at that brain from above. And the front of the brain is on the top, and the bottom of the brain is on the bottom. And you can sort of maybe see that this is sort of brain-shaped. It's brain-shaped enough, right? Those are the regions of the brain you don't care. This is the functional connectivity network that for semantics tasks. That is for tasks that are involving your appreciation of the meaning of language. Now, one of the things I like about this graph is that it's left lateralized, right? And we've all been told language is over here. It isn't exactly, as you can see, but it is left lateralized, right? So the left side of the brain is heavily involved in language, and that's reflected in this graph. But it's also the case, as you can see, that lots of the brain cooperates to allow you to deal with linguistic meaning. This is the emotion graph. What I like about this particular graph, you can see that density in the center. That indicates the connectivity between subcortical parts of the brain. And again, you've probably heard about the subcortical parts of the brain are really important to emotion, and indeed, the graph reflects that. But what's really striking to me about this graph is your brain really is involved in emotion tasks, like pretty much every part of your brain. Now, this isn't surprising when you think about it, but again, it kind of goes against the idea that we've been told that we have these specialized parts. Here is so-called task-driven attention, right? So when you're actually trying to pay attention to something, this is the part of your brain, these are the regions of your brain that are involved in that. It doesn't need all that much of your brain. It's not like emotion. It's charging up the entire brain. But you can see here again, right, we have this sort of right frontal sort of bias, which again, it accords with the story that that's an important part of executive function and attention. But here again, you have multiple parts of the brain cooperating in order to achieve task-driven attention. Okay, so these are the graphs. Graphs are great because you can do all kinds of fun analyses. You can look at the network topology, right? So you can look for where the hubs are. As I showed you in the airline graph, right? Seeing where the density of connections are tells you something about the shape of the graph as a whole. And so you can say, Atlanta is really important to Delta, right? Or Chicago is really important to United, right? And you can do the same thing with these graphs. You can look at parts of the brain that are super important for things. So the anterior insula that I showed you earlier, I won't show you these data, but the anterior insula was interesting to us because it looks to be actually a hub in the brain network. So that looks like one of the things it does for us is it's the switching station that allows us to move between different kinds of functional configurations. You can look for average degree. That's just the number of ingoing or outgoing connections. The thing I want to look at that will get us to our functional connectivity analysis and tell us whether or not indeed the brain is using different functional coalitions under different conditions is a node versus edge overlap analysis. It's super simple. So imagine a really simple brain where the functional connectivity looks like this, where there are 6 regions and you have different kinds of functional connectivity. In a brain that's highly specialized, the one labeled modularity there, in a brain that's highly specialized, you might expect certain regions of the brain to be involved in the red task, right? That's the cooperation under the red task. Maybe that's a visual task. And certain regions of the brain to be involved in the black task. Maybe that's a motor task. And you'd see some overlap between them because, of course, vision and motor control had better be connected or you're in trouble. But for the most part, you wouldn't get very much overlap of either nodes or edges under different conditions. If the brain is doing everything all the time, it'd look more like that holism picture, right? Everything is involved with it all the time in everything. The thing that I am advocating for is that third possibility, which I call neural reuse. And that is that much of the brain is involved in much of your, many tasks much of the time, but when they're involved in different kinds of tasks, they cooperate in different patterns, different partnerships, right? And that's illustrated in that third possibility. So we can do our analysis. To give you some sense of where it's going to come out, here I'm highlighting left precentral gyrus, this is part of your brain right here, and its partners under these various conditions. So those are the partners of left precentral gyrus when you're doing tasks involving linguistic meaning. Those are the partners of that same region of the brain when you're doing tasks involving emotion. And those are the partners of that region of the brain when you're involved in tasks involving task-based attention. Right? So you can see the different fan of connections. Just because it's fun, I'll go back. You can see the different fan of connections and how it changes under different conditions. So that's kind of a qualitative analysis. We can make that analysis quantitative by measuring the amount of node overlap and the amount of edge overlap. So if the brain is modular, you'll get very little node overlap and very little edge overlap. If the brain is holistically organized, you get lots of both. In a brain that's marked by neural reuse, you'll get very little edge overlap, but lots of node overlap under various conditions. Here are the data. Right? So, indeed, it looks like when you look at the brain under different conditions, lots of the parts of the brain are involved across many different conditions. That's the node overlap part. But the way they functionally cooperate is different under each condition. That's the edge overlap part. So you get very little edge overlap and whole bunches of node overlap. So this is an example of what's called large-scale neuromodulation, right? The modulation of neural connections across the entire brain. What's interesting is that there's actually evidence of neuromodulation at multiple spatial scales, from the level of individual neurons all the way up to the level of regions of the brain, and in many different animals. Again, I don't have time to show you all these data, but it's fantastic stuff. We do single neural mapping studies in things like fruit flies and soil nematodes, and we can see that individual neurons are used in multiple tasks, but cooperate with different neurons under different conditions. So this seems to be a super important, fundamental fact about the functional organization of the brain, and of course very different from the kind of things you are typically seeing in the media. Okay, so that's your crash course on brain function. Now we'll talk about why. Why is this, and what it means for how cognition works. So anybody who's been in a basic neuro or a basic psychology class will know this phrase: neurons that fire together wire together. This is so-called Hebbian plasticity, named after the neuroscientist Donald Hebb, who did a lot of work describing this kind of neuroplasticity. This is how your brain, this is one way your brain learns, right? By changing the physical connectivity between neurons in your brain. There's also another kind of neuroplasticity, and this is the stuff I've just been showing you, right? This is called neural reuse. Neural reuse is plasticity at a higher level of organization. This is the process whereby those coalitions are put together. So when you learn a task, you're not only changing the connections between individual neurons, you are also changing the partnerships between larger parts of your brain, and that's neural reuse. So these two kinds of neuroplasticity work together to result in the functional organization of the brain overall. So reuse happens in evolution, right? By the way, there are probably some advanced biology students here. This is meant to represent our phylogenetic history, but you'll notice that this is not a phylogenetic history. This is instead the brains that are easy to find on Google. And because I can't draw, I was limited to that selection. But as an illustration, suppose one of our ancestors has a particular function that allows, a particular set of partnerships that allows it to do, say, some simple visual or motor task. That's likely to be inherited, but the more complex behavior will emerge by taking that part and also sort of adding something to it. And that process repeats itself over the course of evolution until you get to us. And you can see sort of the result of that, right? This is just another network, nodes or graph, nodes and edges, right? So for each cognitive function represented by color here, it uses many different parts of the brain. And each part of the brain, each node there, is used in many different cognitive functions, but they cooperate in different patterns. So that process of neural reuse, putting together those coalitions, happens, as I say, over the course of evolution. It also happens in development. Anything that evolves has to develop, right? We grow into the beings we're meant to become. And so over development, you get a very similar process happening, right? As you learn something, things come online. As you learn something new, one of the things that's going on in learning is you're figuring out which patterns of brain cooperation will result in the best performance on that task. Right? So this happens, as I say, both in evolution and in development. Hebbian plasticity, neural reuse, really important cooperating developmental processes that allow us to learn. And this brings me to the kind of the significance of this for human cognition. We're using the same parts of our brain under many different conditions. That means that parts of your brain that evolved for originally, and that originally developed for things like motor control or sensory awareness, then end up being reused for the kind of human level tasks that we're used to thinking about in terms of human intelligence. Right? So language, logic, mathematics, all these really abstract, fancy things that we do, rely on parts of the brain that are involved in getting you around in the world, in hearing things, in touching things. Right? That should tell us something interesting about the nature of those abstract things. And also tells us something interesting about how cool the brain is, that you can actually use it in this way. So what this means, in part, in fact, this process of reuse results in that thinking is an embodied activity. And thinking involves interacting with the environment. I'll give you some examples. So it turns out motor systems are used in direct support of cognitive processing. And that when you look at things this way, you can understand cognition, thinking, is a series of iterated interactions with the world. So here's a simple experimental example. So that's a button box. Your task, if you were doing this experiment, this is Art Glenberg's work, your task, if you were doing this experiment, is to hold your finger on that red button, and while you're doing that, sentences will appear on a screen in front of you. If the sentence makes sense, you're meant to press the green button. If the sentence doesn't make sense, grammatically or semantically, you're meant to press that yellow button. Now notice that in order to do that, you have to make a motion with your hand away from your body or towards your body. Okay? The sentences are things like, listen to the song, or put a grape in your mouth, or open the drawer, which imply either real or abstract actions, right? So close the drawer implies an action away. Listen to the song implies an action towards, even though it's not a physical action towards. Sell your house, action away. Now, the interesting finding is that things like put a grape in your mouth, you're faster to respond in the canonical, when the direction is the same between what's implied by the sentence and what's implied by your action. And you're slower to respond if you have to make the opposite action. So these things interfere. Close the drawers like this. Joe sang a song to you, right? The same pattern. Even though we're not doing a physical action anymore, there's still an implied direction to the meaning, and it interferes with your actual activity. Sell your house, right? Same sort of pattern, right? So there's an interference between what you want to do with your motor control system and what you're doing with your semantic system, and why? Well, because part of your motor control system is involved in helping you understand things in the world, whether they're physically, whether they involve physical motion or abstract motion. And so here's a brain imaging version of that experiment, where in this case they're just listening to words like lick, pick, and kick. And when you hear the word lick, the part of your brain that's involved in sensory motor control of your tongue gets activated. And when you hear the word pick, the part of your brain that's involved in hand movements gets activated. And when you hear the word kick, part of your brain involved in leg movements gets activated, right? So this tells us probably an interesting story about the nature of semantic meaning, right? That it's grounded in sensory experience, right? This is how it is that high-level meanings, abstract meanings, actually, as it were, mean something to us, right? Because they're activating parts of the brain that are involved in physical experiences. And the result of that is that you get these interference effects, these interesting interference effects. Solving math equations, something similar. This is a cool set of experiments done by David Landy. He thinks, I agree with him, that, and in fact, you probably know this yourself if you think about it, right? When you have to solve an equation, what do? Well, you write it down, you look at it, you figure out which parts need to be moved where, and you move them, and you write out the next version, and you do that until you come to the solution to the equation, right? In other words, what you're doing, and the brain imaging actually shows this, what you're doing is you're treating an equation like a diagram with particular affordances that you can interact with and move things around until you come to the solution. So this so-called abstract task is in fact reusing parts of your brain that are involved in manipulating objects in the world. So this particular experiment is really cool. All he does, so he has these equations, right? And you've got to move the parts around on an iPad. And so you actually move the parts around. This is how you solve these equations. Behind the equations is a subtle moving dots, moving one way or the other, right? Or up and down. The finding is that when the dots are moving in the direction that you need to move your equation parts, you're faster to make the moves, and you do better, right? You make fewer mistakes. When the dots are moving in the opposite direction, you're slower to solve these equations, and you make more mistakes. Now, by typical understanding, this should make no difference to mathematical equation. It's completely irrelevant. And now, it could be that it's just distracting, but then you wouldn't get the differential effect, right? The directionality is what's driving this effect, and it's because you're having to move parts of the equation. You're interacting with stuff in the world. And yeah. Here's another one of David Laney's experiments. I think he's very clever. Which baseball looks closer? I don't know if this effect is working for you, but the first time I saw this slide, it really worked for me, right? The baseball underneath the time sign looks closer. Why is that? Well, because that's the thing you're going to do first. And it turns out this is a ubiquitous phenomenon in perception motor control studies, that the thing you're going to do next, or the object you're going to approach next, looks closer than other objects, even if they're the same distance away. Things like if you're really good at, say, hitting softballs, you judge the softball to be bigger than other people do. If you're really good at making putts in golf, you judge the hole to be bigger than other people do, right? And things you're going to do next look closer. So there's an interaction between your sensory system and your motor system that primes you to do the next thing. This is sort of one of the ways we tell. It's how we can see the so-called affordances in our environment. Affordances are just the way we look at things that we can see what it is we can possibly do. Right? So the doorway affords passage, the floor affords support, the stairs afford climbing, so on. So this is our ability to see things that we can do in the world. The same system that allows us to see what we can do in the world in actual motor control is also involved in things like mathematics. Experiments like this is how we figured that out. Right, so this kind of thing is obviously not predicted by accounts of abstract cognition that treat cognition as abstract. But it's completely consistent with this notion that parts of our brain involved in very simple, low-level motor control tasks, very simple, low-level perceptual tasks, seeing what we can do in the world, being able to act in the world, being able to iterate our interactions with the world, are going to be used and reused under many different conditions. So what it is to learn to do mathematics is actually learning to see what the equations afford, right? What are the actions you can take with those mathematical formulas? The thing that I find super interesting in these experimental studies, and tells us again that we're kind of on the right track here, the better you are at mathematics, let me go back one, right? The better you are at mathematics, right? The students and the experimental participants who are really good at math are more affected by these illusions, if you want to call them illusions, right? So the better you are at mathematics, the more that moving dot thing behind those equations messes you up, right? Students who are really poor at mathematics are trying to sort of go through, okay, what are the rules? What's the next thing I have to do, right? There's the ones who are really fluid with mathematics just see what they have to do, right? They've learned to see the affordances that the equations offer them. And therefore, when you muck with the affordances by putting a moving dot thing behind there, or you do something weird like ask which baseball is closer, right? They're more affected by these manipulations because that's what it is to be good at math, right? In the same way that you'd muck up a good golfer by changing the weight of the golf club or the weight of the golf ball, right? Because they know exactly what they need to do. They see the affordances. Same thing here. If you muck with the spacing of an equation, you muck with the background of an equation, you muck with, right? The people who are really good at math are the ones most affected by that. Again, this sort of modulation of the perception-action coupling system isn't predicted by traditional models of mathematics, but it makes sense in light of the way we evolved and the way we develop to do human-level kinds of tasks. This is just a fancy picture. This actually comes from Tony Chimero. This is a fancy picture of what that is, right? So we have what we do when we engage in the world is so-called sensory motor coupling, right? We adjust ourselves to what's going on in the world, and then we change what's going on in the world because we manipulate things. And so we perceive the affordances, that adjusts the sensory motor coupling, that changes the way our brain is organized. We do something that changes, right? And so this is a process that goes on in real time. And this same process and the same brain areas that are involved in doing this when we're doing physical tasks are the regions that are involved when we're doing language tasks, mathematical tasks, abstract planning, things like that. Right? Preparing to do math. And this, by the way, I think has really profound implications for how we do things like teach mathematics. Right? Preparing to do math is analogous to preparing to take other actions. And it modulates the sensory motor coupling in the same way. Right? So abstract cognition is just a way of using and reusing parts of our brain involved in getting around in the world and manipulating our environment in the service of high-level, complex kinds of tasks. So thinking with your brain is also thinking with your hands, because you're reusing the parts of your brain involved in moving your hands, in moving objects, in manipulating parts of your environment. And that's how it is that you learn to think high-level thoughts. And I will stop there and be happy to take questions.
So I'm a senior from Bonchek, and I was wondering, you said that we perceive the baseballs with the multiplication signs to be closer because we know that's supposed to come first. Have there been tests on like young children who haven't learned that rule, and if so, does it just not affect them at all, or how much does it affect them?
Yeah, no, so yes, if you haven't learned the rule, you do not have this effect. The way they did this is they've done tests with children, but also with college students, but they give them unfamiliar symbols. And with unfamiliar symbols, there's no effect. Then they teach them that the symbols mean things like multiplication or division or addition or subtraction. And as they come to learn and get used to that, then the effect comes back, right? And David has also done mean things, like manipulate the spacing of equations. And so he gets a classroom of I think, like fourth graders learning basic arithmetic kind of skills. And they space the equations so that they're consistent with the order of precedence or inconsistent with the order of precedence. The kids where they space it so it's consistent learn a lot faster, right? Because it's easier now to see how to do the math. And the kids that they're mucking with and doing the spacing inconsistently, it takes them longer to learn. What's interesting in this particular case is then if you switch them, right? If you give the kids who learned with the consistent spacing, inconsistent spacing, they start making more mistakes. And the kids that learned with the inconsistent spacing, takes them longer to learn, but when you give them the consistent spacing, it makes no difference. Right? So you're changing the way they see the equation. So you can make kids learn faster, but it also then makes them even more sensitive to these manipulations.
I'm Pablo Hennig in biology. For what I remember learning is a lot of this localization of functions in the brain was deduced not from before functional imaging from people who had brain damage in different parts of the brain and that affected only certain capacities. How does that fit with your model?
Yeah, thank you. So in reality, any given brain injury affects multiple things. And we've always known that. If you look at the clinical studies, you have a particular, maybe a stroke or a blow to the head or whatever the issue is. You lose multiple capacities, right? Your motor control is affected, your language is affected, and various things. Because we had a theory whereby parts of the brain were specialized for different things, those results were generally interpreted as being a problem with the specificity of the lesion, right? People said, oh, well, if only the lesion were more specific, you would have had a more focal deficit. But because natural lesions are really messy, we're seeing multiple things. So it's very interesting, and this has happened multiple times in the history of neuroscience. In this case, we sort of switched the data and the noise, right? We thought the multiple deficits were noise, and we had to read into the focal thing to get the data. But in fact, the opposite was true, right? That actually, those were the data. And as you say, so there's actually a very interesting project, I think it's run out of the University of Minnesota. Since we have been unfortunately involved in multiple wars and conflicts recently, there are a lot of soldiers with head injuries. And they've been mapping all the deficits that are involved in all the head injuries in the folks coming out of the armed services. And what they find is consistent with what I've been showing you here. That parts of the brain, they have different effects, but there are always multiple effects. And this is called lesion function mapping. And it's useful for therapy, and it's useful for other reasons, but it's also interesting scientifically.
So I had a question just about the brain imaging that you showed earlier.
Sure.
There were actually gray sections to the brain. I didn't quite understand what that meant. Did that mean that they just weren't active? Because I thought this was showing total diversity.
Yeah, thank you for that. No, the gray sections were just, I didn't have data. So when the parts of the brain were gray,
Yeah.
those were sections for which I didn't have data. So, yeah.
Hi, I'm Eric Talbody from Computer Science. I was curious or what's understood about what mechanisms mediate these different coalitions. Like when there's very specific regions, you can say, perception kind of decides what task you're doing, and then it decides who to bring online. But if everyone is doing everything, who's deciding which coalition to bring on to the task?
Yeah. So there isn't anybody who's deciding. We don't have a homunculus in there actually driving the switches. What seems to be the case, and we're at the very beginning stages of this kind of thinking and this kind of research. What seems to be the case, those functional fingerprints represent the functional dispositions of individual parts of the brain, what it is they like to engage in, basically. So when the environment affords particular kinds of possibilities for engagement, right, that information gets spread throughout basically the whole brain. But only the parts that like to engage with those things, that are sort of attracted, that have the disposition that match those conditions, end up being activated by those conditions. And so the different coalitions come about, and it turns out that those networks that I showed you are so-called assortative, right? So the networks are generally composed of regions with similar functional fingerprints. And so what seems to happen is the conditions, and of course internal conditions as well as external, right? The conditions drive the activation of regions of the brain that then become active together because the conditions are right they match their dispositions. Does that make sense? Yeah. So that's it's kind of and this is true, I think, once you start thinking of the brain this way. There's the internal mechanism, but there's also, as it were, an external mechanism, right? We're driven to be engaged in the environment, and the properties of the environment sort of call online various parts of the brain, the ones that are particularly good at or particularly disposed to engage in whatever opportunities are afforded in the moment. The low-level biological details we do not know. There's all kinds of interesting biological mechanisms here, including things like genetic expression. So it turns out that genetic expression can turn synapses on and off in real time. It turns out that there's something called spine motility. So dendritic spine. So at a synapse, there's a dendrite and an axon, and the spines can actually twitch away or towards the synapse, right? So that's protein folding issue, right? So you get genetic expression, you get a protein folding issue, and you can just turn the synapse on and off by that. Now, how that's coordinated, who knows? But there are interesting mechanisms that permit this really rapid changing of configurations.
This has really been a fascinating talk. I'm Scott Lerner. I'm in Italian and French and comparative literature. I wonder if you're aware of implications for your research about the relationship between conscious and unconscious thought and or memory.
One of the areas of psychology and neuroscience that I've studiously avoided is consciousness. Because it just seems too So I don't think I do have anything for you on that in particular. One thing I will say about memory, though, we tend to think of memory as another abstract store, as if we're just taking experience and putting it and storing it in some kind of veridical sense. If you're thinking in terms of an action-oriented brain, the kind of brain that I'm describing here, the so-called blue-collar brain, the brain that works with its hands, that's probably not a good way to think about memory. Memory is, in fact, a way of learning what things to do different or the same in the future. Right? So memory is all about figuring out how to act differently. And when you then look at the so-called mistakes we make in memory, like when we recall things that didn't actually happen but we think happened, they're almost always adjustments to the memory that better serve its action relevance. Right? So we're storing things in a form that makes it better for us to act with or act upon. And we shouldn't expect memories to be veridical. They're reconstructions in the service of action. So there's definitely that implication of this work for the nature of memory. And ask me in 25 years about consciousness.
Arnie Feldman in the math department. I had a curious reaction to your problem. I saw the word baseball. I looked at the baseballs and I was dimly aware there were numbers, and I thought the plus sign looked better. I wonder if that's the kind of thing you can take into account in experiments, or would the experiment have been phrased differently, like work this problem and then, or something? Because I didn't realize there was a math problem there. I just saw these baseballs I was supposed to work on.
What kind of math do you do?
Algebra, and I'm actually extremely good at arithmetic because I'm so old that there weren't calculators when I was in college.
Well, here I'm going to quote Brian Scholl, who says that we have a term for individual differences in cognitive psychology. We call that noise. There are clearly individual differences here. And how your particular experience of that and your particular background would map onto that modulation of perception would be sort of interesting, right? We could gather a bunch of people of your cohort and expertise and see if you get a consistent sort of experience of that equation or not. The dirty secret of psychology, not really a secret. Almost all of our experiments are done with college-age students. Right? Because that's what's available to us. And it's certainly going to be the case that as we study different populations, we're going to get different results for some of these things.
Clark Madden, I'm in no way a plant or longtime friend of yours. I wonder what, if anything, the interaction of the principles that you articulate of functional fingerprints and reuse suggest about the evolutionary future of the brain.
The evolutionary future, well, there are two things to say about that, I think. So one is that one of the things that's super important about neural reuse is that it gives us immense flexibility. Right? Because we can put together pieces of the brain in all kinds of different configurations. Right? So that suggests that we will be very, very able to adjust to very changed circumstances. Right? In other words, think about the combinatorial issue here. Right? If one part of the brain was dedicated to one thing, and say you had 1,000 regions of the brain, you could do 1,000 things. But if instead those same 1,000 regions can be put together in different functional coalitions, you can do 1,000 by 1,000 by, right? So our behavioral flexibility then becomes immense as a result of this. Evolutionarily, I think that the reason this capacity developed, why it was preserved, is because of exactly of that. Because it allows us to use neural tissue, which is very metabolically expensive, in the most efficient way possible. Right? However, so that gives us a really flexible mechanism. One thing that is worth pointing out, however, and mothers in the audience will be well aware of this, our heads can't get any bigger. Right? So we may well be, unless there are very large physical changes to our bodies, we may well be at a point where we can't add capacity to the brain, right? Because we can't make the neurons faster, because the only way to do that is to make them bigger, and then they won't fit, or to myelinate them better, and then they won't fit. And we can't make the connections any denser, because then they won't fit. Right? So the only way to get it would be to grow the brain bigger, and then we can't be born. And so, again, unless there are concerted developmental and evolutionary changes for the entire body, our brain is about the size it's going to be for the foreseeable future. However, even if, say, the environment changes dramatically, which it seems likely to do, because we have this developmental mechanism of changing the coalitions of the brain, we have immense behavioral flexibility.
My name is Gian Zlupko. I'm an SPM major, and in about 30 minutes or so, I'm presenting to Anderson a topic of my own research that is very similar to his. So I thought I'd use this as an opportunity to
You're
ask
not
for
getting
an
out of
answer
your presentation.
to my thesis. No, but the question I had when you were discussing some of the abstract tasks that we do is whether or not we've identified certain tasks as requiring a greater degree of embodiment than others. So, for example, mathematics versus maybe language processing. Or articulation?
That's an interesting question. I don't know about a greater degree of embodiment. I'm not even sure how I would measure that. I have to think about what that would mean. It is interesting, and maybe this is related to what you're interested in. Tasks that emerge later in development or that evolved later, so tasks like language and mathematics, use more and more widely scattered parts of the brain than tasks that emerge earlier, like vision or audition, that use fewer and more tightly grouped parts of the brain. And this appears to be because the later in development, the later in evolution you are, the more parts of the brain are already doing stuff that's interesting and useful. And so the more options you have for putting pieces together, right? So that's not greater embodiment exactly, but it does show that there's this increasing sort of scatter and increasing number of parts that are used in these later developing tasks.
Hi, I'm Amy Molnix. I work in the faculty center. So I think I have two questions if we have time. The first one is, how do we know that the images that you're collecting, which are on different individuals, actually represent brain functions that are consistently mapped to the same locations?
Okay, so the answer to question one is they don't always, but we would do various things. So typically what you do in an imaging experiment is you look at what parts of the brain are active under various experimental conditions in an individual. Then you map that individual's brain to a template that makes everybody the same, right? And then you average. That's probably going to introduce, there are individual differences, but when you look at the way the templates work and how things are averaged, you get something like, it varies a little bit, but something like 80% agreement between brains. So there are individual differences, but they're not huge. And that seems to be because we're mostly solving the same kinds of problems, and early in development, early developing parts of the brain are pretty stereotyped in the way they develop. And so we end up with sort of the same elements of the brain early on, and then we end up going through the same educational system. And so the same coalitions make sense to most of us most of the time. And so there are differences, but they're not huge. So there's a very prominent scientist at MIT who doesn't believe me about this stuff. And she had the same question. She goes, well, that's just an artifact of the averaging. It's not really true. So she started doing things just with individuals. And turns out, although she doesn't admit it, but if you look at her data, it turns out you still got the overlaps, even in individuals, right? They look a little different than they look, but the principle still seems to hold, even when you're looking at single individuals and not averaging across.
Okay, my second question is, so when you map a part of a brain and you see it active in an image, you are looking at a population of cells.
Correct.
So do we have an understanding of whether the connections, like whether a group of cells in a particular area subspecialize? So I might be in this area, but I am going to be the cell that connects to the emotion, and that cell is going to be the one that connects to that. So,
Yes.
so that's part A, but part B is if it's not the cell, are they different spines on a sort of, are they different subcellular parts?
I'm reminded now you're a biologist. Neuroimaging, yes, we're looking at many, many, many thousands of cells all at once. And there is an issue is, okay, well maybe again, when you get lower than that, then we get specialization. So the way we look at that, we look at other animals where we can do single cell recordings, and it turns out when we do single cell recordings in other animals, you get the same kind of multiplicity of function. In humans, we do really mean stuff like the following. This is Art Glenberg again. He says, here's a pile of coffee beans, like 1,000 coffee beans. I want you to move this pile to that place, one bean at a time. So you do that. So you're moving your arm away from yourself over and over and over again. And then he has you do a language comprehension task. And what this does is it actually fatigues individual cells, right? Individual neural cells, because you're doing the same thing over and over again. And when you do that, then you're actually worse at understanding sentences involving action away from yourself. Although your understanding of action towards yourself is intact, right? So that looks again like, oh, no, it's actually the individual cells, right, that are being used for both tasks. And so you're fatiguing them by moving these coffee beans, and it makes you harder to understand particular kinds of language.
Okay, thank you.
So I think this is going to be the last question. Yeah.
Hi, Mike. It was a great talk. Matt Butterfield, Department of Music. I'm still stuck on the baseballs.
Okay.
So things that we're going to do next look closer. And what I'm wondering is there an analog in sound perception?
Oh. I have no idea. But it's an interesting question. Yeah. I really, that's it. I'm a scientist, so I admit ignorance.
Well,
I don't know of any data about that. Interesting question.
this is the last Common Hour talk for this semester. What a wonderful way to end. Thank you so much.
Thank you.